Welcome back again to Biology Boost Up. Today in this video tutorial session we will study about the process of translation or the mechanism of protein synthesis. So now let's start. Before going to study about the mechanism of translation, we should study about the activation process of amino acid and the attachment of active amino acid to a specific tRNA. At first, after the transcription, all types of RNAs are come to the cytoplasm from the nucleus. On the cytoplasm, by the help of ribosome, the RNA synthesized protein by the process of peptide bond formation among the amino acid, and the process is known as translation. The ingredients are needed for this mechanism are mRNA, tRNA, amino acids and some protein factors. The process of translation occurs in the cytoplasm of both kind of cell. Next, the mechanism of amino acid activation. The activation of amino acid occurs through their carboxyl group or COOH group. Each amino acid is catalyzed by its own specific activating enzyme, that is amino acyl tRNA synthetase, and ATP here helps to formation of amino acyl adenylate or active amino acid. A covalent linkage is formed between 5' phosphate group of ATP and carboxyl end of amino acid, and this molecule remain attached with this enzyme. Now the process of attachment of active amino acid to a tRNA. Amino acyl synthetase and ATP makes the amino acid active and then helps to formation of amino acid tRNA complex. This synthetase has two sites, one for recognition to a specific amino acid and another for recognition of specific tRNA. This enzyme catalyzes the formation of acyl bond between amino acyl adenylate to 3' terminal adenine nucleotide of tRNA. This ester bond is formed between 3' OH tRNA and carboxyl group of amino acid. This is the diagram of mechanism of amino acid activation as well as the attachment of active amino acid to a specific tRNA. Now talking about the initiation of translation. At first, by the help of initiation factor, that is IF1 and IF3, the 30S subunit first attached with 5' end of mRNA, which is known as SD sequence or Sine-Dalgano sequence. The P site of 30S subunit of ribosome remain at the site of initiation codon, that is AUG. As a result, the IF3, IF1 and 30S mRNA together form 30S initiation complex. Now, after the formation of this complex, the FMate tRNA amino acyl tRNA attached with AUG codon at the P site position. Now, by the help of initiation factor 2 and GTP, the 50S subunit comes and combined with the 30S initiation complex and form 70S initiation complex, and here GTP converted or transformed into GDP and organic phosphate. Next, the elongation of translation. During this elongation process, the elongation factor or EFTU and EFTS and GTP helps to binding of amino acyl tRNA to the A site of ribosome according to the codon of mRNA. Now, by the help of peptidal transferase enzyme, peptide bond is formed between two amino acids which are attached to the A site and P site of the ribosome respectively. Now, during this process, the ribosome moves 3' direction of mRNA and the deacylated tRNA transfer from P site to the E site of ribosome. At the same time, amino acyl tRNA which attached at the A site transfer from the A site to the P site also. As a result, now the A site comes to a new codon of mRNA. Then another new amino acyl tRNA or amino acid attached tRNA attached to the A site and new peptide bond is formed with previous amino acid which now present in P site. This process occurs multiple times. Now, the process of termination of translation. During the elongation, when ribosome moves towards the 3' end of mRNA and when a termination codon is translocated into the A site, then the ribosome, instead of binding with a complex containing amino acid, binds to a peptide released factor. There are 3 release factors are present inside the cell, that is RF1, RF2 and RF3. The RF1 identifies the termination codon, that is UAG and UAA, and the RF2 identifies the termination codon, that is UAA and UGA. The functions of RF3 binds GTP and seems to stimulate the action of RF1 and RF2. It is also known as stimulating factor or S factor. The release factor interact with peptidal transferase, causing hydrolysis of the polypeptide chain at P site. This resultant cleavage and release of chain from the tRNA molecule at the P site of ribosome. The ribosome then dissociates into 3S and 50S. Thanks for watching my video. Please like, share and subscribe the channel.